Okay.
Looks like it's going.
It's good?
Yeah,
Okay.
good to go.
Okay, so Alex, do you want to start and I continue introduction, or how do? Or
You can go just in case, because this is more of a public place and I don't know if I'm going to be interrupted. So
okay, good. So it's a pleasure to introduce Jena Johnson from University of Michigan. And just before I do introduction, couple announcements. We put some information relevant to resources for people, for scientists escaping from Eastern Europe. So feel free to either add your name if you wish to the list of labs that accept them or able to host them, or spread the word about these resources. Second, we will continue next week and we will have a presentation by JP Duda. It will go back to organic geochemistry, probably mostly on Neoproterozoic, but maybe he will take a broader approach. And will be a couple changes in the schedule later at the beginning of April and May. So at the beginning of April, and Alex will put it next week on a schedule, but we will have a second talk from Liz Turner. She was willing to help us and step in and replace the speaker, and she will talk about Sturtian record from Congo Craton, from Democratic Republic of Congo. And when we're still figuring out, and we will have a replacement in early May as well. So stay tuned and check the schedule. So now it's a pleasure to introduce Jena Johnson from University of Michigan Ann Arbor. She did undergraduate in geobiology in Brown University and followed with PhD in geobiology at Caltech, which she got in 2015. And I believe she studied with Woody Fisher. And at that time, during her PhD, she focused on photosynthesis and the rise of atmospheric oxygen using manganese record from South Africa, specifically from Kuhas and Hotazel formations. And during that time, he started to recognize importance of laboratory work, experimental work, and study also field analogs. And she continued with postdoc at the University of Colorado in Boulder between 2015 and 2017, where she got exposed to experimental work. And she now an assistant professor in Earth and Environmental Sciences at the University of Michigan, starting in January 2018. And she focused on understanding evolution of life and environments as recorded in chemical signals. And I know specifically, or one of the projects she works on laboratory sort of production of greenalite, which is, I guess, relevant to the talk she gave today. And with this, I pass it to Jena.
All right. Well, thank you again for inviting me, and thank you all for being here. So today, I'm excited to tell you all about some new insights we have into the chemistry and potentially biology of the early ocean through experiments exploring these products of biogeochemical processes that may have occurred in the ancient ocean. And I'll get to the greenalite too. So just to start broadly, as we look back over geologic time, we have a great fossil record, less great fossil record, and then we really don't have a clear fossil record before around 2 billion years ago. But we do have these iron formations, which are iron-rich marine sediments that we find in both Archean and Proterozoic deep ocean or deeper ocean settings. So these are some examples of iron formations, and they are just potentially incredible and informative about the early ocean, because they are deposits from the deeper ocean without any or much materials from continents. And these strange rocks seem to have been widespread in the ancient ocean, and they contain around 30% iron and 50% or more silica. You can see the abundance of this silica as all these white nodules and lenses throughout these different photos. So Archean iron formations are predominantly thinly laminated or banded, you can see here. So we call them banded iron formations, or BIFs. They are composed of finely crystalline materials on a microscale, implying that these minerals started as small chemical precipitates. This is particularly exciting because chemical precipitates from the ocean can record the chemistry and potentially biologically driven chemical reactions of the ancient ocean. So before we get into these, I want to just back up and ask, how does an ocean actually make one of these iron formations? And let's focus on the BIFs, the banded iron formations, which dominated the Archean marine record. So it's actually a better question to begin with, is why the modern ocean doesn't deposit banded iron formations. And let's start with where iron comes from. So iron is introduced to the surface environment as reduced ferrous iron 2+, from hydrothermal vents like mid-ocean ridges, and from weathering igneous rocks in the continents. But in today's world, we have 21% oxygen in our atmosphere, as we all really appreciate. And this oxygen will react with reduced ferrous iron to make solid iron 3+ precipitates. We can see this in the red iron oxide-rich soils developing on basalt, like this example in Madagascar, or iron oxidizing to orange iron oxides around hydrothermal vents, like this example at the Luihi Seamount. So the modern ocean doesn't really get the iron released from these iron sources. Instead, iron is largely trapped in iron oxides around the hydrothermal vents or on land in the soils and sediments. So the first thing, step one, to make a BIF in the ocean is a lack of oxygen. So iron stays as soluble ferrous iron 2+ and can be concentrated in the ocean. In fact, the deposition of BIFs and other observations led geologists to realize very early on that the Archean atmosphere didn't have much, if any, oxygen, allowing this ferrous iron to build up in the ocean. But now, to form preserved BIFs in our rock record, we need to transport that iron into the sediments. So we need to precipitate the iron. So to precipitate this iron, the critical question is, what was the mechanism of iron precipitation? And only then can we get at if this is recording signals of early life. And before we understand this mechanism of how iron got so enriched in the sediments, we need to understand what the original precipitates were. The problem is that BIFs are billions of years old, and like all ancient rocks, they've seen multiple episodes of hydrothermal fluids and alteration. We can see the effects of these fluids in cross-cutting regions of alteration, both reduction fronts like here and oxidation fronts like here and here. And on a microscale, iron formations are extremely complex with many different iron-bearing minerals, including oxidized and reduced iron in disequilibrium. This degree of remineralization in ancient iron formations, and knowing that iron can undergo many transformations under various fluid and metamorphic regimes, has made it really difficult to assess what the original iron precipitate was. And without knowing how these sediments got so enriched in iron, it's impossible to draw conclusions about the original ocean chemistry or the microbial ecosystem. The best way to determine what the primary minerals were in these iron formations is by looking at early diagenetic chert, like these chert nodules. And that's because we can tell this chert grew in the sediments early after deposition and before the sediments were compacted, because we see the sediments deform around that chert nodule, reflecting that first the chert mineralized and then the surrounding sediments lost their pore water during compaction. And as this early chert silicified, it could incorporate and encase any original minerals that were present and preserve the primary minerals far better than the surrounding iron-rich sediments. A common inclusion found in cherts is hematite, which has suggested that first ferric hydroxides, like here, were the earliest precipitates, and then they aged into hematite during diagenesis. Now, this conclusion that iron oxides were the most primary BIF mineral has led to several hypotheses involving both microbial and abiotic iron oxidation, leading to the formation of BIFs. For example, low levels of oxygen from oxygenic photosynthesis being used to oxidize iron, or iron being oxidized via iron oxidizing photosynthesis, or UV-induced oxidation of iron, all generally leading to iron oxides being deposited and forming the precursors to BIFs. However, in the last few years, a group led by Rasmussen in Australia has suggested that these iron oxides could be secondary, based on observations of cross-cutting oxidation fronts in this BIF chert and secondary veins mineralized by hematite. They found that the chert exposed to these oxidizing fluid fronts has inclusions of hematite, like here, that were actually replacements of iron silicate granules. And the best preserved chert has inclusions of iron silicate nanoparticles. And there's also cavities that could facilitate fluid flow. So from this multi-scale set of observations, Rasmussen et al. Has been arguing that iron silicates were the most primary particle, going against the previous paradigm of primary iron oxides. So what does this tell us? What do these primary iron silicates signify? Again, to pinpoint the mechanism of iron precipitation, we need to know still more of the exact chemistry and mineralogy of these early silicates. So we need to ask, what is the most primary silicate mineralogy? And this is important because silicate clays is still a big group of minerals. They all have layered structures with hydroxide layers and silica tetrahedron layers that come together to have this characteristic layer spacing. So which one of these was the most primary silicate mineral in the ancient Archean Ocean? So here, to answer this question, we collaborated with the Rasmussen group and looked at some of their best preserved cherts from both Western Australia and South Africa to investigate what the exact mineralogy and redox state of the iron is. Like the Rasmussen group, we observed that these quartz euhedral crystals with abundant nanoparticle inclusions were cemented by quartz that's free of nanoparticles. And then using a variety of methods, we first characterized the mineralogy of these inclusions. So the clearest method was transmission electron microscopy, or TEM imaging. So here we could see the individual particles that were really a variety of sizes, but mostly 500 nanometers or less. And as this example shows, this TEM high resolution imaging identified these particles as having approximately 7 angstrom spacing, which is consistent with greenalite. And as we mapped iron, we saw that iron was really only in these particles. And in fact, using electron dispersive spectroscopy, or energy dispersive spectroscopy, we could see that the major cation in these particles appears to be iron, confirming that these mineral inclusions are greenalite. Now, we also wanted to measure the redox state of iron in these particles at the scale of individual particles to see whether redox reactions could still play a role in the precipitation of these minerals. So we took TEM sections of chert, the Canadian light source synchrotron, to scanning transmission X-ray spectral microscopy, or the STXM, where we can focus only on the iron and measure the iron redox state at a 30 nanometer scale. This technique measures the iron L edge, which distinguishes between iron 2+ and iron 3+. And we used a calibration based on the ratio of the iron 2-3 peak to quantify the amount of iron 3 present. This was developed by Bordell et al. In 2013. And then we rastered the beam throughout the nanoparticles at these key energies to create an iron redox map at the nanoscale. So this was wonderful because we could pair TEM imaging with this synchrotron-based redox analysis to look at the iron redox state across a variety of particle textures. For example, this cluster of particles, shown in the white arrow, looks well preserved and encased in silica, as compared to this small particle or this cluster of particles that are associated with cavities that could facilitate fluid flow. So now I'm going to overlay the iron redox state, where the iron 3+ content is plotted in deep red, being all iron 3+, to 0 iron 3+ in purple, and all these colors in between. And we can see that the better preserved cluster of greenalite nanoparticles have iron redox states that vary from around 10 to 20% iron 3, while the more altered greenalite has an iron redox state that varies from 20 to upwards of 50 or 60% iron 3. And 3 other well-preserved chert samples with nanoparticles had extremely similar distributions, with these well-encased particles having an iron 3+ content of around 10 to 20%, while particles that appear more recrystallized or associated with cavities have a higher iron 3+ content. Okay, so the most primary particles are greenalite, with 10 to 20% iron 3, but dominated by iron 2. What does this characterization imply about the iron precipitation mechanism, and thus the chemistry and biology of the early ocean? We have a couple hypotheses here. So one is that abiotically, ferrous iron and silica could just be saturating and forming greenalite particles that are fully reduced. However, this would suggest that the ferric iron we detected in these exquisitely preserved greenalite particles is all secondary. And it contradicts the iron isotopic record, which points to iron cycling in iron formations. Another idea is that iron could be oxidized to iron 3+. This again could happen either abiotically by UV rays or biotically via small amounts of oxygen from oxygenic photosynthesis or iron oxidizing photosynthesis. And this iron 3+ could trigger the precipitation of an iron 2-3 silicate in a silica-rich ocean. Now, there have been several studies, mostly out of the Kepler lab, that have looked at iron oxidation in high silica fluids. And this work has shown that iron 3 oxides, here, not silicates, are this terminal product of microbial iron oxidation, with the silica getting absorbed or incorporated into the iron oxides. So we actually already know that if iron is completely oxidized to iron 3, the product is oxides, not silicates. but we think there was plenty of iron 2+ around in the ocean. So maybe the iron silicates and iron formations were catalyzed by iron oxidation that's just a partial iron oxidation. So we need to investigate if partial iron oxidation in a ferruginous environment could in fact trigger an iron 2,3 silicate to form. Or another hypothesis from Matai Halevi's group is that partial oxidation like this could first form an iron 2,3 intermediate phase, like a green rust, which is a mixed iron 2,3 hydroxyanion salt, which then stabilizes in the presence of silica as a silicate. Or perhaps iron 3 oxyhydroxides were formed, but then they were reduced in the sediments, perhaps by iron reducing microbes, to form low iron 2 or low iron 3 greenalite and silica. This type of iron reduction in sediments is really common and performed by iron reducing microbes. So we've been working to test all of these hypotheses. First, a grad student working with me, Isaac Hinz, has led the charge to investigate whether either ferrous iron interacting with silica or partial iron oxidation in high silica water forms greenalite. So in this project, we set up an artificial Archean seawater and added 1 millimolar silica and 1 millimolar iron, with the iron either completely ferrous or containing 5% ferric iron to sort of simulate partial iron oxidation. We sampled the room temperature precipitates and precipitates after they were hydrothermally aged to promote a crystallization, sort of simulating diagenesis. As previous research from Nick Tosca's group has suggested, we observed that when it's completely ferruginous or reduced iron, pH is a critical control on iron silicate precipitation in anoxic water, with higher pH promoting iron silica precipitation. So what we saw was at and above pH 7.5, iron-rich silicates form. These are mostly poorly ordered, but we were able to measure the crystal structure on a few of these particles, and they seem to have spacing similar to greenalite. However, in addition to the problems that I already mentioned surrounding the lack of the ferric iron in these precipitates, this ferrous system's pH requirement for being at or above pH 7.5 is concerning, because the Archean ocean pH has been modeled to be around 6.5 to 7, although potentially was as high as 7.5. And Chris Sanson-Tutton's estimate of this neutral pH at this time is consistent with another model, which similarly predicted this pH range of around 6.3 to 7.2. So maybe iron oxidation could change this pH requirement? What we saw was yes. In our experiments with 5% ferric iron, precipitation occurred at all of our tested pHs, including pH 6.5 and 7, where there were no observable precipitates in ferrous experiments. Now, these lower pH solids at room temperature seem to be mainly amorphous iron silica co-precipitates. But Isaac did find rare layered structures with a greenalite-like structure. Interestingly, we saw nothing similar to green rust in these initial low temperature precipitates. After we did our kind of simulated diagenesis 150-degree crystallization step, we saw abundant minerals, including ferric oxides, magnetite, green rust, and also many layered structures, shown here. that again had the spacing and elemental chemistry of a greenalite. So testing both ferrous iron and mixtures of ferrous and ferric iron showed that we could form greenalite either in high pH water with this ferrous system, or partial iron oxidation could be a mechanism to form greenalite at pHs below 7.5, although the ferric iron additions did form some of these other phases as well. So more recently, Isaac has been exploring what phases are produced when the iron is oxidized just partially in situ. And here he's been bubbling in trace amounts of oxygen, 50 ppm, for 90 minutes into an anoxic Archean-like environment with this simulated iron and silica-rich seawater. And here, what we see is this green precipitate forms. And again, by TEM imaging, we see a much more homogeneous assemblage of mostly proto-iron silicates that we're recognizing now as this kind of hair-like curling sheets that form bundles of a couple layers. And ferrozine measurements show that these are about half iron 2 and half iron 3. So mixed iron 2-3, we think proto-silicates that when we can measure the spacing, do again seem to have this spacing of around 7 to 8, 7.5 angstroms, just like greenalite. If we age these precipitates for a week at 80 degrees Celsius, we see beautiful silicate crystals form with greenalite-like spacing and elemental chemistry of an iron-rich silicate. So in situ, partial iron oxidation does form abundant greenalite, and it's a much more homogeneous assemblage with just a few other minor phases. So overall, both of these experiment sets show that partial iron oxidation could be a mechanism to form greenalite. But we also wanted to test this alternative end member hypothesis that perhaps greenalite could form from initial iron 3 oxides, then getting microbially reduced in the presence of silica. And this project was led by grad student Chrissy Nims. So here we started with the ferric hydroxide ferrihydrite in the presence or absence of silica to really explore what this silica presence does to the secondary minerals. And so she added a model iron-reducing bacterium and excess organic carbon, so the bacteria could theoretically reduce all of the iron oxide. Then we let these experiments proceed for over 4 months, well beyond any visible evidence of continuing reduction. The products we saw are quite interesting. Calcium carbonate precipitated regardless of the condition, so even in the controls, which just showed that our system was very saturated in calcium carbonate. And we also saw needles of the ferric oxide, goethite, form in both the presence and absence of silica, although it was much more abundant when silica was not present. Comparing the silica present absent experiments, what we saw in the presence of silica was that both our mineral characterization and ferrozine results indicated that biologically produced ferrous iron was precipitated alongside silica in iron silica co-precipitates, which again form these kind of curling bundles that look like these proto-silicates we've been seeing in other experiments. So these could be precursors to greenalite. Just like what we saw from the partial iron oxidation, these precipitates, these iron silica co-precipitates, are again amorphous, and they are rich in iron and silicon, just like greenalite would be. So we think these are potential precursors of greenalite. So as we test this alternative hypothesis that the microbial reduction of ferric oxides in the presence of silica could form silicates, we find this is potentially another way to form greenalite, although we still see other phases like ferric oxides persisting. So in conclusion, there's strong evidence that low iron 3 greenalite was an early mineral in BIF deposits. And we're now experimentally testing potential mechanisms for how this low iron 3 greenalite can form. And we've observed that ferric iron does trigger the formation of greenalite alongside other phases, suggesting that low iron 3 greenalite we see in BIFs could be recording signals of iron oxidation, maybe iron oxidizing life. But we also see that iron reducing bacteria can biomineralize iron silica co-precipitates that may be precursor iron silicates. So this suggests that perhaps the iron silicates could be recording iron reducing life, or maybe both is going on. So we still have more to explore here, but we're getting some exciting results about what this low iron 3 greenalite may be recording about early chemistry and also early life. And with that, I'd like to acknowledge my funding sources and our lab group, and I'll end back on my conclusion slide to take any questions. I think we have plenty of time for questions, so happy to chat further about any of this. And again, thank you.
Thank you, Ina. And as we did before, we should start first with relatively simple questions and broad questions relevant to whole audience. And when, with time, we progress to more specific questions relevant to smaller group. So feel free to write your question in chat or unmute yourself and ask questions.
That was great, Jena. I've got a very simple comment, actually, mainly. I'm really enjoying what you're doing with this, particularly the redox state of the iron. It's just fascinating at this level. But there's a definition problem here. I think that I heard you slip into the old Canfield idea of ferruginous ocean for the Archean. Ferruginous is actually a Latin word. It means brown. I think what you really mean, like ferruginous hawk, for example, Falco ferrugineus, you know, it's a common classical word, which has a meaning which is the exact opposite of what you're talking about. You're talking about a ferrous ocean, or you could call it a Canfield ocean too. Both are good, but it's not an ocean that has a lot of Fe3+.
No, I definitely mean iron 2+.
Right.
I wasn't aware of that as a definition problem. Thank you.
Just etymology. But great talk. I'm really enjoying what you're doing.
Thanks.
So Ina, it's great that you show different pathways, but how we now can test between them in terms of which one maybe in different time in your history operated? Like, did you think to combine it with iron isotopes or with scavenging of trace elements, or with something that would give you a better idea about potential precursor? Like, was it a complete iron oxidation, or was it partial iron oxidation?
Yeah, I actually have a collaboration that's really led by Andy Hurd,
Mhm.
one of Nicholas Dafa's students, looking at iron isotope signals in greenalite versus iron oxides. So I think iron isotopes are a great way to approach this. I think my next step is first to look at what do iron oxidizing phototrophs produce, but then also to try to get this kind of dual system of having a partially oxidized product from these types of microbes or maybe low iron, low oxygen bubbled systems, and then feeding them to our iron reducers to sort of see what mineralogically that produces. Because there are other minerals like siderite or iron-rich carbonates that really suggest microbial iron reduction was occurring. So I still want to find a way to produce that and confirm that these kind of experimental systems do show that as well.
So like if we compare polyproterazoic granular iron formations and Archean banded iron formations, in terms of iron isotopes, clearly Archean iron formation have a partial iron oxidation. And I don't do you think it has implication for this model that require sort of complete iron oxide precursor, or would it be still consistent with it?
Yeah, I think the iron isotope folks are really consistent with what we're proposing with partial iron oxidation. And I think another thing to test is the iron redox state of some of these products from both oxidation and reduction experiments. But yeah, I'm also very interested in the post-rise of oxygen proterozoic formations and trying to better characterize the iron silicates there and kind of do this side-by-side comparison. But it's tricky, and I mean, with such ancient sediments, it's always really hard to be definitive about which pathway is the right one. And as much as I have ideas to keep going and trying to test, I'm not sure if we'll ever be able to say that these were certainly from only partial iron oxidation versus then they were secondarily processed by iron reducers. But I think partial iron oxidation is probably the way to at least get the minerals there.
Great. We might come back, but I think you have one, I guess, question in the chat from Karthik Murami Karnan. Does the presence of greenalite vary across oxygenation events and the first instances of cyanobacteria? And Karthik, maybe you can unmute yourself. By oxygenation event, do you mean through Earth history, or maybe you can clarify what you mean? Okay. Unless maybe Ian, does a question make sense to you? Can you reply to it?
Yeah, I don't actually see the question, but
Oh, sorry, it was a direct question to me.
From what you said, I can talk a little bit about, I mean, well, not really we, the Rasmussen group has characterized greenalite starting back over 3 billion years ago until 2.4 or 5 billion years ago. And then Letitia Bregman, and I'm sure others too, are characterizing greenalite in iron formations that are post-rise of oxygen. So I think this is a phase that's important throughout a large part of Earth history. Does it vary? Yeah, well, there's a big question of when are the first instances of cyanobacteria. The greenalite that I characterized the redox state of was really from pretty much 2.5 to 2.6 billion years ago, because those are the best preserved. So I don't know if there is a change in the greenalite going deeper time, if it gets more reduced. And we also haven't characterized the redox state of the greenalite post-rise of oxygen.
Okay, thanks. And Andreas Kapler has a question, so maybe you can unmute yourself and ask.
Yes, hi Jena, very nice talk, thank you. And I have a question regarding the iron reduction experiments. You mentioned that you saw calcium carbonate in the absence of silica. Did you also see it in the presence of silica? And if yes, is this related to the bicarbonate concentration that you used? Did you use a bicarbonate buffer?
Yes, I think that's certainly it. I think we used 10 millimolar bicarb as a buffer. I get lost trying to remember all my students' experimental details, but I think it was 10 millimolar. And
okay, no
yeah, that was definitely much higher than a similar set of experiments that was out of Mike Tice's lab that didn't start with, I think, much of any bicarb and just let the bicarb evolve from the organic oxidation. And so they saw siderite, whereas we saw calcium carbonate. And so we put in the discussion this whole thing about, well, why are we seeing something different here? It's probably the just starting saturation state of calcite.
Did you also see any siderite or not at all?
No.
Interesting.
Yeah, I think it was just the ratio of calcium to
Silicate and that thing. Yeah. Mm-hmm.
Yeah, I mean, it was 10 millimolar calcium when it was present, and then iron was only 1 millimolar or so, and so I think it just got swamped.
Mm-hmm. Perfect. Thank you very much.
I'd love to make siderite.
Thank you.
So Lee Camp raised the hand, so unmute and please ask question.
Thanks, Andre. Thanks. That's really, really exciting. I love the experiments and the advanced characterization approach. It's really cool. You're our resident manganese expert, and I'm wondering, have you done any experiments with manganese? Is there any co-precipitation of manganese under any of these conditions? And if not, and I think in general, the low manganese content of BIFs, what is the manganese cycle, Archean?
Yeah, great question. I love going back to manganese. I do have a student working on a manganese experimental project, but we haven't included any manganese in these kind of BIF simulation experiments. But it would be an interesting thing to add to just see whether it does get incorporated at all. I mean, I think that the lack of much manganese at all in BIFs up until the rise of oxygen shows that oxygen was not a factor in forming BIFs, and it's much more likely iron oxidizing phototrophs, which is why that's the next thing I want to test, because I think we need an iron specific mechanism. UV oxidation could also be another factor that would be just selective for iron.
Yeah, the pH change driver for green rust formation, for example, in an anoxic environment's always intrigued me. So I wonder about how strong those pH requirements, those model pH requirements are. I think they depend on, approximations around calcite saturation, because you got PCO2, right? But you need more than that to constrain the carbonate system. And so, I can't remember what all the constraints were on those, but they probably, thinking of, conjuring up Dick Holland saturated with calcium carbonate type, and then you need to know the calcium concentration. So the precipitation of ferrous hydroxide and decomposition into into magnetite, and then in the presence of silica, irons, mixed valence silicates always was interesting to me as a productivity driven, whether it's photoferrotrophy or just pH increase due to planktonic or photosynthesis, was always of interest to me as a driver here. It looks like ferrous iron oxidation is your trigger, however that's driven here, I suppose, right?
That's what I would say. I mean, the Rasmussen group and the Tosca group also have a very different view, where they actually think that the higher pH might have been happening in hydrothermal vent fluids, and then greenalite got dispersed around. I think the iron isotope record is probably our best argument against that. But yeah, I agree that the models of the Archean pH are tentative.
Okay,
Great.
thanks.
So we have a question from Greg Ritalik, and when we will go to Caroline Dreher.
Hello again.
Yeah, Greg, maybe you can ask question.
Yeah, I typed it in. I've been working in the Pilbara, and there's a wonderful little 3.6 billion year old banded iron formation in the Panorama Formation, which Bolhar used rare earth elements to determine was actually fresh water. I agree that most of the big ones, the Fortescue ones, are marine. What do you think about that? And how could you tell whether it was marine or freshwater? Are there, your experiments are all marine salt concentrations, right?
Yeah, I wasn't aware of this BIF. That could be freshwater, but I think in some ways not that much changes. We have done some experiments without the high salts, and we don't see massive differences.
Pretty similar. Yeah. Well, the best modern analog is actually freshwater crater lakes too, as you're probably aware.
Yeah, I visited some of those in Mexico, hoping to find a good.
Oh, sweet. Yeah. And what did you find? Well, that's a whole different study, I suppose.
They were high silica, but they didn't go anoxic enough. Yeah.
And the minerals, no greenalite, right? A lot of the hematite in the Mexican lakes. Yeah. No, it's intriguing. Thanks again.
Thanks. Carolyn?
Caroline, can you unmute yourself and ask?
Yes. So I'm a PhD student with Andreas Kappler, and I'm currently in the middle of cycling experiments trying to form BIF minerals. I'm doing my cycling experiments with cyanobacteria and Chivanella, and currently I am at the end of my first oxidation state, and what I see especially in the higher iron concentrations is that I in fact do have a partial iron oxidation. And my next step now is to look at the mineralogy, and I was also thinking about that maybe greenalite formed. And so I'm super excited to hear that this is, that you found out something similar. And so my experimental conditions were in general pretty similar. So I think it might be worth chatting about experimental setups in the future. It's really, really interesting what you found out.
Yeah, that's awesome. I can't wait to see more of your work.
Yeah, so not really a question, just a quick checkup that I'm finding out hopefully similar things.
Yeah, feel free to email me. I'm happy to chat more about anything.
Okay. Alex, you want to ask a question? Go ahead.
Yeah. So I guess I didn't know, is it Jena or Jena?
It's Jena, actually.
Jena? Okay, okay. So I had a couple thoughts. My first one I want to attack is from the perspective of iron isotopes. And so the iron isotope composition of these there's a couple of the two sort of hypotheses one being iron oxidation was happening, and then the minerals transitioned to a greenalite mineral. So that doesn't necessarily preclude the concept that oxidation was having its influence over the iron isotope composition of these. But the alternative that you can do it by reducing the oxides in the sediment and then generating greenalite, do you think that actually agrees with the iron isotope record that we find?
Oof. If you do iron isotopes, I would defer to you or Andre about that.
Oh, okay.
I don't actually, I'm not an iron isotope person. I just talk
okay.
to them sometimes and
yeah, I was wondering, since you were working with Andy Hurt and Nick Duffus, I was wondering what your thoughts were, but that's okay. My next one is more of an broad scale.
Oxidation. I think the partial iron oxidation is more consistent with what's observed, as I was talking about with Andre, but I would defer to the actual iron isotope people.
Okay, got it. Yeah, it's confusing. It's not. So I was just curious your intuition. But my other thought is more about just general ocean chemistry through time. I know you were focusing on the BIFs now, but after the Great Oxidation event, we start finding these granular iron formations in abundance, and there's a consistency in the mineralogy in a sense that you still have mostly iron, and your iron oxides and stuff. But travel further into the future past, into the Phanerozoic, you have Phanerozoic ironstones, which their major difference when it comes to their iron silicates is they have aluminum. And I'm wondering what you think about that. Why aren't we having aluminum in even maybe these granular iron formations in the Precambrian?
Yeah, it's a great question. I mean, I think the lack of aluminum is really revealing and really indicates that these are water column precipitates in both the case of BIFs and granular iron formations. And maybe the granular ones are then either building up in the sediments as to granules or rolling around in the sediments, but they don't seem to be getting much terrestrial input. Whereas, yeah, in the Phanerozoic, it seems like a very different mechanism that's much more influenced by weathering. But I haven't studied those Phanerozoic ironstones, so I wouldn't speculate too much about that.
Okay, yeah, I mean, it perplexes me because you have basically oolitic granular iron in both situations. That just their mineralogy and the layering is, I mean, you have silica and iron oxide layers in your GIFs, and you have aluminosilicate and iron oxide layering in your oolitic ironstones. So it's just curious to me. Thank you very much.
Yeah, great questions. I'd love to talk to you more about this, but I don't want to sound like an authority when I'm not.
That's okay.
Yeah, we should chat at some point. Let's see, we have another hand raised and I cannot locate. Muammar, please unmute yourself and ask your question.
Can you hear me speaking?
Oh, yeah, it got better.
Yeah, okay. Yeah, it's a really nice talk. So I have a question about how to detect greenalite. So obviously these are nanoparticle minerals. Can you detect greenalite via XRD, or do you need more sensitive methods like TEM or Raman or something else? How can one, if you find this, if you make this mineral in an experiment, how can we not miss it, basically?
Yeah, great question. Sadly, because I have a Raman in my lab, Raman is not a great technique for characterizing greenalite, especially in these older rocks where the greenalite is embedded in microcrystalline quartz. So we actually just see a signal of the quartz. We found, and this is in the 2018 paper, that XRD, we could see really small peaks of greenalite, but it was again really dominated by quartz. So that's where by SEM, we could see there were these inclusions, but again, not really characterize them. TEM, after years of trying, I found that it is the best technique to characterize whether or not you have greenalite and what the spacing is. And the amazing thing about TEM is that there's also these kind of many different measurements that we can make there, both on the electron diffraction, on energy dispersive spectroscopy, just actually high resolution spacing data. So that's really powerful. I think Andreas could, Kapler could like tell more about the Moss-Bauer way of trying to characterize greenalite versus other iron minerals. So I wouldn't say TEM is the only way, but I just think it's a really great technique to do this.
Okay, great. We ran out of raised hands and questions, so we can transition to more specific discussions. So if people have anything more specific to ask, feel free to unmute yourself and ask.
And I know.
Can I ask another
so there is one hand raised from Daryl. Yeah, go ahead.
Yes, a general question that applies to all of you guys working on iron formations. One of the things that is really noticeable is they're so well laminated.
Yeah.
The processes you're talking about look like a big, big chemical beaker, and I would have thought they would have resulted in a much more amorphous, massive deposit. So what do you think is the significance of those laminae?
Well, I don't think my beakers or my bottles are really accurately simulating an ocean. So we're just going for kind of mineralogy match. I think the lamination is largely due to this gravitational settling, and then also compaction within the sediments.
Well, that would give us a uniform, if it was simply occurring in an ocean, it would give us a uniform rain of material. It wouldn't give lamination. Do you see any evidence that might be related to Milankovitch cycles or tidal cycles or anything like that? Has anybody done that?
There's been some earlier work speculating about whether these could be tidal or Milankovitch. I don't know if Paul wants to chime in here.
There's a paper on it. I know I saw something.
Was a paper by Lanting probably 4 or 5 years ago, where
you're muted.
They calculated and related to Milankovitch cycles, and also Brian Krepes and other people argued about contourites that might be explained with laminations.
Nucleation inhibition tends naturally to lead to dumps of precipitate. So I think there's a natural tendency for chemical sediments to come out in pulses.
Okay.
Alex Trendle, thought long and hard about the lamination in BIF, and various forcing forces were investigated without a clear resolution, I think. But it may be a natural tendency because of the need for oversaturation to incite precipitation. Lee Kump probably could have.
Well, I just put something in the chat there about the possibility of diagenetic separation, and into the resulting iron-rich and silica-rich phases from this initial precipitate. Something else that's been proposed.
Yeah, I guess it depends what scale of lamination, because I think Paul's probably correct on the really fine sub-millimeter laminations is more just because of episodic deposition. But then there's the larger scale kind of layering that may be more diagenetic.
Yeah, I guess, you know, the question is the driver for the silica-rich component of that. Then I think you have a mechanism for the iron silicate part, but how to get the chert from this in the absence of, you know, biogenic chert precipitation has always been an issue, right? You know, especially tied to an upwelling model, which seems to go the wrong way unless the temperature structure of the ocean was upside down.
Well, I guess Woody Fisher, you know, proposed silica absorption onto ferrihydrite particles, but that implies iron oxide as the primary phase.
And one aspect that you need to consider is when the iron oxides are biogenic, then there is almost no silica absorption to the oxides. That means you can separate the oxides from the silicates by including a biological oxidation mechanism. And then when you consider a temperature dependence of microbes, already small changes in temperature would allow the microbes to be more active at slightly higher temperatures. And then you reach oversaturation of silicate. And if you lower the temperature, you precipitate a silicate. We did some of these experiments with phototrophic iron oxidizers, and it works perfectly well. You can separate the iron from the silicate, the iron from the silicate, just by running higher, lower temperatures. Just temperature differences by 10 degrees Celsius are sufficient already.
And Brian Kerpez also has nice observations suggesting that a lot of silicification happens just at the sediment water interface as sort of a secondary chert growth that I've seen a lot of the kind of similar textures.
Hard to change the temperature of the seawater because the wind blows. I live by the ocean and every day it's the same.
So why biologically precipitated iron oxides would not scavenge silica? Is it because we're at sediment water interface, or?
No, the reason is that the biomass, they coat the iron oxides, and the iron oxides are usually positively charged. And then you bind all the negatively charged biomass, and this rejects the silicate. Okay. You can just coat the oxides, or the oxides when they are associated with all the biomass, the EPS and the cells. This is why you separate the silicate.
Thank you.
Carolyn Dreher had her hand up for a while now.
Yeah, Carolyn.
Yeah, I have a question about the experimental setup. If I remember correctly, you used 1 millimolar of silica and 1 millimolar of iron too, right?
Yes.
In most of your experiments. How much do you think does greenalite formation depend on the iron-silica ratio? So would you expect a big change if you increase or decrease one of those concentrations?
It's a great question.
I know.
we actually started with higher silica concentrations of more like 2 to 2.2 millimolar. And the products we got were a lot more amorphous. And then it seemed like some recent sort of modeling and constraints suggested that maybe silica wasn't quite as high. And then also Nick Tosca's group is arguing that iron was much higher than we think, or we used to think. So it could have been in the millimolar range. So I think it's something that we have to still explore, that ratio, but at least small differences don't seem to make a huge change in our products.
Okay. Yeah, because I'm also just wondering, because when I started to prepare the artificial seawater medium with high silica values, I had the problem that a lot of silica didn't go in solution, so a lot precipitated directly because I think of oversaturation. So did you manage to find a way to keep the high silica values in solution to start those experiments?
Well, by going to 1 millimolar, that seemed to work better.
Okay.
You were up in the 2s. It did, once something precipitated, then there was just that surface nucleation effect and a lot of silica splash out.
Okay, thank you.
Yeah.
Okay. Any questions, comments for Jena? Okay, looks like people are satisfied. Thanks, Jena. It was great talk and well balanced on this obviously very controversial topic. So with Alex,
discussion. I've really
Alex and I will connect to you maybe next week. Thank you, everyone. And next week we will have talk on organic geochemistry, I guess, on Neoproterozoic from JP Duda. And Alex will announce it later this week. Talk to you later. Thanks, everyone. Bye.
Thanks, Jena. Congratulations on getting to Ann Arbor.
Thank you. Thanks for your feedback.
Thank you. Bye.